Welcome everybody to this Memory Decoding Journal Club. This is, in a sense, an encore to one that I did in April that was about grid cells. Here we're going to be looking at the system in the medial entorhinal cortex that provides input to those grid cells, and attempt to learn a little bit about what that circuit might be based on data from stimulation of specific cells and recording from other cells. I'm actually not extremely happy with this choice of paper now that I'm presenting it, because I was hoping to present something that would give more insight into a model of grid cell driving and how activity of grid cells changes as you move around in an environment. And for that, I would probably have had to reach back to an earlier paper, an older paper. There's a paper from the Hasselmo lab that does a fairly good job trying to describe that, but that's all the way from 2014. And I felt a little bit strange going all the way back to 2014 to present a paper from then. So we'll just see how well this paper does. This is synaptic interactions between stellate cells and parvalbumin interneurons in layer 2 of the medial entorhinal cortex are organized at the scale of grid cell clusters. And just a quick look back at what we did last time. So not last time, but back in April. That time I presented the paper, Grid Cells Accurately Track Movement During Path Integration-Based Navigation Despite Switching Reference Frames. In this paper, there were two very important insights that came out of that paper. The first is that it's possible to build a decoder where if you translate grid cell firing by changing the coordinate system that you're working with translated onto a torus, that you could do that with a decoder and you would get a signal where the grid cells no longer appear to be firing as grids, but instead they're firing as attractors on a torus, and where grid cells that have similar grid activation are also nearby on that torus. And then the principal insight from that paper was that grid cell activity appears not to be one static map, but instead re-anchors. So as you're entering a new environment, you have one set of grid cell firing that is based on features in that environment. And then if a mouse reaches a lever, then the grid cell firing re-anchors to that lever and becomes relative to that location. You can still get the same bump. You can get bumps on attractors, that remains, but the firing is now relative to a different aspect of the environment. And then, of course, at the end of that paper, we were left with the question, well, this bump attractor, this activity on the torus, how does it shift around from one place to another? And the input to grid cells is essential for that. So this paper is about the input to grid cells. I just heard a tone. Is that someone asking a question? Yes, Ken.
Yeah, it's me. I'm sorry. I shouldn't ask a question this soon. So I read through the paper, but maybe I missed this. Aren't the stellate cells that they talk about in the paper grid cells?
They are also grid cells, yes. You see grid cell-like behavior on both pyramidal cells and stellate cells in medial entorhinal cortex.
Okay, so what do you mean by saying that this is talking about the input to grid cells?
That's my interpretation, in the sense that the pyramidal cells in that area also exhibit grid cell activity, and so you could consider this as input too, or this could be talking about the circuit that is processing input to the medial entorhinal cortex and generating grid cell-like activity by using the recurrent activity between stellate cells and interneurons. So in that sense, maybe input isn't the right word for it, but it's the, instead of looking at the grid cell activation, this is the precursor. It's the how do you get grid cell-like activation. That's what it seemed like to me. anyway.
So I just want to, I mean, everything is complicated. I just want to make sure. So if I think about this in terms of the stellate cells that they're talking about are grid cells, and the inhibitory interneurons that they talk about are setting up the attractor dynamics. Is that have I lost the plot if I think about it that way, or is that just a simplistic way of thinking about it?
No, you haven't lost the plot. This is the part of the circuit that it's about. And then here the question is more about the architecture of the arrangement of those stellate cells and how you end up with clusters and modules so that you can look at the generation of different types of grid cell patterns.
Okay.
And yeah, so it's about this aspect. Yeah. So this is really a paper that's about that architecture and trying to determine what's connected to what and how things are clustering. The primary objective of the paper was how could excitatory-inhibitory interactions determine location representations in grid cells, and the approach they're using to get insight into that architecture is they're applying optogenetic stimulation, and they're doing patch clamp recording. And as we'll see, they do that in both directions, from stellate cells to the interneurons and from interneurons to stellate cells. So critical determinants of the interactions include spatial extent, over which given presynaptic neurons influence their targets, and whether connectivity is specific to subsets of neurons within the potential target population. Now, patch clamping is the gold standard, but it's still very hard to do that on very many neurons to assess with high throughput. But they try to do patch clamping here. Grid cells in the medial entorhinal cortex have an anatomical organization with features similar to the columnar organization of sensory cortices, and nearby grid cells have a similar spatial scale and orientation maintained across superficial and deep cell layers. For grid cells at different dorsoventral locations, so that's top down from, say, back to belly, the scale and orientation of their firing fields varies, with larger scales found in the more ventral neurons, so the neurons towards the belly. This variability arises from discrete groups of grid cells that have firing fields with similar scale and orientation, and they form functional modules. Individual modules can be partitioned further into cell clusters, with each cluster containing topographically oriented ordered grid cells encoding the full range of possible spatial phasings. So the question then is, what are the mechanisms that establish the organization of synaptic interactions at these anatomical scales? In medial entorhinal cortex layer 2, the interactions between excitatory stellate cells and inhibitory parvalbumin positive interneurons, so the PB plus interneurons, as I'll be calling them from here on out, they've received attention as a possible substrate for the emergence of grid firing fields. Both stellate and pyramidal cells in this layer have grid firing fields, in answer to your previous question. The interactions between stellate cells appear to be primarily via parvalbumin plus interneurons, and models based on that connectivity are able to account for grid firing as well as local theta nested gamma frequency network activity. That goes way back to all that modeling that I once tried to explain about MEC and hippocampal activity, which I won't get into right now. But okay, so electron microscopy reconstructions could constrain the potential connectivity, but the authors here say that those are limited because they don't reveal functional interactions. Now, if we had good structure to function, that could be very different. Oh, actually, I wasn't quite done yet. Sorry. The blue part here is about their main findings. I kind of wanted to give you those up front. Let me just take a quick sip of water. So they find that extensive axonal projections support dense but incomplete stellate parvalbumin connections. The spatial organization of synaptic interactions consists in is consistent with local modules that are defined by excitatory-inhibitory connections. Dense, specific excitatory-inhibitory interactions operate at the scale of grid cell clusters, the closely located neuron pairs that receive common inputs. And indirect interactions coordinate at the scale of grid cell modules, where there are intensive interactions within clusters of those neurons and weaker interactions between clusters. So overall, this gives some insight into the functional dynamics of the entorhinal cortex. Okay, let's look at the anatomy first. In figure 1, they're taking a peek at the structure of stellate cells and trying to define where most of the axonal and dendritic fiber is. In figures 1A and 1B, you can see that the stellate cells here were reconstructed following patch clamp recordings from sagittal, that's dorsoventral there, and horizontal brain slice. And then they also, as you can see next to that in these little recordings on the extra panel, they established the electrophysiological profile of those recorded neurons. In C and D, what they did is they overlaid the axons that have been reconstructed and then plotted out the length to angle plots. And you can see that there's a dominance of the extent of the fiber in layer 2. But you can, I think you can kind of see that just by looking at it. In figures 1E, you've got the left and right panes here. And on the left, this is the total length of axons, and on the right, total length of dendrites, and then it's plotted for each of the layers. And you can see that there's definitely a, the distribution of the axons and dendrites is kind of similar, very much in the first 2 layers. And we go to figure 1F. This is where they are showing that the axon length in layers 1 to 3 doesn't really predict the length in L4 to 6. It's kind of all over the place. And then in figure 1G, if you look at the maximum extent of L1 to 3 axons, that was greater than the dendrites along the dorsoventral axis, but similar along the medial-lateral axis. And the result is really that these extensive axonal substrates for the stellate cells would be able to, if you look at the extent here, coordinate other neurons located along both the medial-lateral and dorsoventral axis in L1-2 of MEC, because that's where it's most extensive. Next, they investigate if the extensive axonal arborization that we see here leads to functional coordination of targets of stellate cells separated by similar distances within L2. Let's move on to that. And this is where they first start to use or show the protocol that they have for the optical activation and recording by patch clamp. What they're doing here is activating stellate cells, which they're setting it up so that channel rhodopsin 2 is conditional on the presence of Cre, M cherry is conditional on the presence of Flp. And what you can see is that the stellate cells are labeled in green, and parvalbumin cells, the interneurons are labeled in red. And then in figure B, what we have here is a wide field optical stimulation, and they're ramping up the intensity of that stimulation. They're giving an optical ramp, and you see the response coming out of that. And then over here on the right, they're comparing 2 different stellate cells, one in blue, the other in green. And one of the things you can observe already here is that they're asynchronous. They don't fall on exactly the same time points as they're being activated by that ramp. So here in C, we have the setup for the wide field optical activation and the recording with patch clamp from parvalbumin interneurons. We're showing 3 interneurons here, which they then show again in D, where in D we can see the excitatory postsynaptic currents that are being generated in those interneurons as a response of the activation of stellate cells. And there is a strong correlation in PV for those PV neurons that are activated by the same stellate cells. And Here we see 3 is different. It's not being activated by the same stellate cells. And you can also see that here in the correlation and lag plots, but that is different. Then in E, we have on the top here convolution of the EPSC waveforms by stellate cells in the parvalbumin interneurons, and on the bottom we have how they're showing that they're going through a series of stimulations where they are comparing different amounts of stellate cell overlap while maintaining the same total input to parvalbumin interneurons. And that's to show what kind of a difference it will make if cells are being activated by maybe the same stellate cells or not by the same stellate cells, using this overlap as a way to test that hypothesis that you can figure out where your activity is coming from, even when we don't have the very focused activation yet. So in these results, you can see that the cross correlation of postsynaptic current, as we see here in F, that they can index the common synaptic input from the upstream neurons activated by a ramp-like stimulus. Moving on to figure 3. Now we're going to be looking at the organization of correlated input. They're recording from pairs of cells, and they're recording from pairs of cells at different distances. So they're looking at the effect that difference between different PV cells makes. And in A here on the upper side, we can see simultaneously recorded excitatory postsynaptic currents from pairs of PV interneurons. This is actually in A and in B. And here we have a correlated pair, an uncorrelated pair. And as we look at the lower panel here, we're seeing measures of the maximal correlation coefficient and the lag variance between these two responses to the asynchronous activation of those stellate cells. And in A, the input is from a shared population, whereas in B, this is from 2 different populations of stellate cells. So you can see there's a stronger correlation here and less lag, and that's kind of what you would expect. Figure C and D are a zoom into these little red windows here. So this what looks like a line is actually a tiny window. So blowing that up here, and you can look at the individual current traces and cross correlation within the peak cross correlation windows for A and for B. And there's a very clear example of synchronization versus asynchronization or asynchronous activity in here. So they're just setting up their method, really. They're trying to prove out that they can determine what is driving these interneurons based on the activity that they're seeing, and that they can do this by testing out the responses of interneurons at different distances. And here in E and F, this is where we're going to take a look at that distance dependence. So we see a difference. So we see small separation of the interneurons and larger separation of interneurons. And then they're plotting out the peak cross correlation coefficient and the variance lag in F. And they're checking that for dorsoventral axis versus mediolateral axis. And what we can see here is that when they're more correlated, they tend to have smaller distances between the cells. They found a result for 35 out of 71 recorded pairs that received coordinated synaptic input from stellate cells. And the asynchronous wide field activation that they did in this experiment for stellate cells apparently can be used to identify pairs of parvalbumin interneurons that receive coordinated synaptic input. The coordination by the stellate cells depends on the extent and direction of the separation between those interneurons. And pairs of interneurons that receive coordinated synaptic input from the stellate cells were typically separated by distances up to about 500 micrometers along the dorsoventral axis and up to 900 micrometers along the mediolateral axis. Is that? Let me just check. Sorry. Is that a hand up? Oh, it is.
Sorry, me again.
Maybe I should go back. There we go.
Yeah. So just a quick question. The dots that are labeled in yellow and blue, correlated and uncorrelated PV neurons. So I guess what I'm confused by is there's the y-axis is correlation and the color is correlation. So why don't those track like perfectly? There's a different way they're correlated. Yeah.
So this actually comes up in their results later. Even though you have a lot of cells that are only a certain distance apart from one another, you have a different correlation outcome depending on whether or not these cells were receiving input from the same stellate cells. So what they're ultimately telling you is that, yeah, even if they're closer together, you still find a lot of uncorrelated cells all throughout at every distance.
So for example, there's a blue dot on top of a yellow dot in figure E. And
Yeah,
I don't
one of those.
see why. So that blue dot seems more correlated than the yellow dot. The yellow dot is labeled correlated.
Okay, so the peak cross-correlation coefficient isn't the only measure here. They've already done the comparison by comparing EPSCs across the entire time interval and just looking at the cross-correlation over time.
Okay.
So this isn't just about the peak. The peak is what they're only showing in ENF, but they have already classified cells as being more correlated or not correlated.
Okay. Okay. Thank you.
Yeah. All right. Moving on to the next step in their procedure. This is figure 4. We're looking at the effect of synaptic inhibition. So this is to test if the coordination of the parvalbumin interneurons by stellate cells involves the recruitment of intermediary neurons that coordinate the spike timing by stellate cells. For that, they've evaluated the effect of blocking fast inhibitory responses mediated by GABA A receptors. And they're using gabazine to do that. So in figure 4A, they're just showing the blocking of fast inhibition by gabazine, the effect that has. And then in figures B and C, we're comparing control and gabazine. Control here shown as gray and gabazine as green in at two different distances. We've got 101 micrometers, we've got 388 micrometers. And they're showing that, so they're showing this as a function of time since the onset of light in stellate cells. And they're comparing proximate cell bodies and distant cell bodies. And they find that the blocking fast inhibition has an effect on synchronization distance. In 4D, they go a bit further. They've got heat maps here at different distances, dorsoventral, mediolateral, and again, compare control and gabazine. And overall, again, they're showing the effect that gabazine shuts off long distance synchronization. In 4E, they're showing again that the gabazine affects all but the shortest distances, and there's a strong dorsoventral effect. So for pairs of these parvalbumin interneurons that receive coordinated input in control conditions that were separated by less than 150 micrometers, when the GABA A receptors were blocked, their responses to ramp activation of stellate cells were similar. But by contrast, for the interneurons separated by more than 150 micrometers, the cross-correlation peak mean was typically reduced, and the cross-correlation lag variance was increased. And in many cases, the responses to ramp activation of stellate cells were no longer consistent with the PV interneurons receiving coordinated input. So that just disappeared, that coordination, suggesting that their coordination in control conditions involved inhibitory interactions between stellate cells. That suggests that coordination of PV interneurons involves multiple mechanisms with distinct anatomical organization, where there's a direct electrical synapse interactions that are restricted to PV interneurons separated by less than 100 micrometers. And I believe they did some extra tests demonstrating that these are probably electrical synapses Partly this is from other work, but also in the supplementary material that I didn't go into here. And then the coordination of PV interneurons by stellate cells is more extensive and is both direct and via additional inhibitory pathways, which appear to increase the extent of coordination. So now they're switching to a slightly different protocol, no longer using the wide field optical. So that's because they're trying to corroborate their findings by doing focal activation of stellate cells. So if the focal light pulses asynchronously activate restricted populations of stellate cells, then simultaneous postsynaptic responses in pairs of interneurons will identify inputs from the same stellate cell. That's the idea, at least. So in A, we see the setup. They've got this grid of blue dots in the background indicating these focal points for the laser that's used for the optogenetic stimulations that they're carrying out. And at the same time, they're doing patch clamp recordings, of course, in the PV cells. Now in B, we can see an example of this for two different cells, 1A and 1B, where at the blue bar, this is when the light, the optogenetic stimulation is being carried out, light is on, and then these red bars are indicating separate events that are happening. And in C, this is giving us an indication of how they carry out the temporal binning for the results that they're showing us. But we can skip straight on to these heat maps. I think this is the more interesting insight. So what they did here is that they're looking at the current amplitudes at stimulation locations, different stimulation locations for that focal activation for cells 1A and 1B. And where there's a high correlation, it's indicative of a shared input stellate cells. So then their examination of these maps, one of the things that rolled out of that is that there are strong correlations and anti-correlations that seem to be associated with clear borders between the locations, the regions that are indicated here. And then in F, that the correlation as function of distance, we have here again different distances between cells, are shown here, and the red ones are the ones that are above chance. Those are not above chance down here. So high correlation probabilities indicate common synaptic input only for PV interneurons within about 200 micrometers in sagittal slices, 400 micrometers in horizontal slices. So the result here is that this data shows that focal laser stimulation of the channel rhodopsin expressing presynaptic neurons in combination with simultaneous recordings from postsynaptic neurons is able to assess the extent to which pairs of neurons receive common input. And that supports the conclusion from the previous wide field activation experiments that the connections from stellate cells to parvalbumin interneurons are locally structured and specific. This is the last part of the same figure. I couldn't put all of that on one slide, and they kind of move on as well here to a different part of this. If grid cell clusters result from locally organized connectivity of the neural circuits in medial entorhinal cortex, we'd expect to find a corresponding organization of functional stellate cells to parvalbumin interneuron connections. Stimulus locations that activate pairs of stellate cells should either be similar if they're both part of the same functional zone, or distinct if they're each members of different zones. That's that hard boundary there. Alternatively, similarity between stimulus locations for pairs of stellate cells should have a random distribution. That's if that hypothesis doesn't hold. So they built maps for the responses of individual parvalbumin interneurons across all stimulation sites. And they then took these correlations of heat maps of parvalbumin cells, and they compared those with shuffled heat map correlations. And when you compare the original with the shuffled data, it falls way outside of that distribution. You can see it's pretty clear difference here. And then in figure H, we see the Pearson correlation by distance, and the distance along the bottom. And we see that it's much stronger at shorter distance. and that pairs with strong spatial correlations typically have higher temporal input correlations, whereas pairs with low temporal input correlations also have low spatial input correlations. So this suggests a structural functional organization of input from stellate cells to the PV interneurons and provides evidence for anatomical boundaries between populations of stellate cells, and suggests that for pairs of parvalbumin interneurons that receive common input from stellate cells, the common connectivity is not all to all, but arises from distinct subsets of stellate cells. All right, so how does it work in reverse? How about the feedback from parvalbumin interneurons to stellate cells, the reverse direction? In the next part of the paper, they focused on that, and they start here, of course, with the anatomical analysis, just like they did in the first part of the paper. And they're looking specifically at parvalbumin interneurons with axons that are restricted to layer 2. You can see that restriction pretty clearly here, especially when they overlay all the axons on top of one another, it becomes a very clear indicator. And axonal output from most PV interneurons with cell bodies in the L2, layer 2 of the medial entorhinal cortex, is indeed restricted within the layer 2. That's in agreement with previous observations. And you can show that these axonal projections are quite extensive, often approaching 1,000 micrometers. So it's like we're redoing some of the experiments from before. Right now, we're just doing them in the opposite direction. And this next step, again, is like the previous figure. So this is to address the extent to which the organization of inhibition from parvalbumin interneurons to stellate cells mirrors that of the coordination in the opposite direction. They targeted expression of channel rhodopsin to parvalbumin interneurons. And in this case, now they're recording from pairs of stellate cells in that layer 2. So in figure A, they're showing the protocol for the optogenetic stimulation. And in figure B, they're just showing us recording in cells in layer 2. And in figure C, this is again like in the previous part of the paper, we're seeing the iPSCs, in this case, not EPSCs, inhibitory. So it's iPSCs recorded in stellate cells for the ramp of optical stimulation as that goes up. And yeah, this is what you would expect. In contrast to the responses of the parvalbumin interneurons to activation of stellate cells, this opposite direction, the PVN-driven synaptic currents recorded from stellate cells, were relatively sparse and often of large amplitude. A subset of the pairs of stellate cells showed responses to activation of PV interneurons that were highly correlated with little variation in their lag, indicating that these pairs shared input from the same PV interneuron. So this is the same basic protocol, we've just reversed everything. So here we have an example of strong correlation. Then in D and E, we're again looking at the peak cross-correlation and lag as a function of the distance between cells, but in this case, the distance between recorded stellate cells, but also in E between stellate cells and pyramidal neurons. So here's a comparison of those different cell types. The cell bodies of pairs receiving common input were typically separated by less than 300 micrometers along dorsoventral and mediolateral axes. And pairs of neurons that did not demonstrate correlated input were found at all distances tested, once again, indicating that close proximity is not sufficient for stellate cells to receive input from the same PV interneurons. The pyramidal cells in layer 2 of the MEC have a topographical organization that's distinct from the stellate cells and receive distinct inhibitory input from CCK positive basket cells. And you can see the relationship between stellate cells and the low correlation with pyramidal neurons here. This is indicating that the pyramidal neurons indeed receive input from a distinct population of interneurons. All right, we're getting closer to the end here. So what the authors did here is they developed new strategies for assessing common input to pairs of downstream neurons from the genetically defined upstream populations. And they were using simultaneous paired recordings combined with optogenetic activation, as we saw. And here are some insights about the circuit organization in medial entorhinal cortex layer 2. The stellate cells and PV plus interneurons provide common synaptic input to one another, but connectivity is incomplete with a correlation coefficient between 0.4 and 0.6, suggesting partially overlapping rather than all-to-all connectivity. Many nearby cell pairs share no common input, implying membership in distinct subnetworks. Indirect inhibitory interactions can extend to the spatial range of coordination, particularly along the dorsoventral axis. And SEs and pyramidal cells do not share common inhibitory input from PV interneurons, consistent with those two cell types receiving inhibition from distinct interneuron populations. The authors then sketched out a what I would call fairly simple model of the idea of how these clusters operate and how the modules operate. And it's based on a sketch of a three-level hierarchy where you use where gap junctions are responsible for the coordination of PV interneurons within about 100 micrometers, and the stellate cell to PV interneuron modules operate at up to 250 micrometers, and where longer range connections between the modules could be mediated by non-PV interneurons. They, for example, mention layer 1 interneurons that have fairly extensive axons that could be responsible for that, but they don't really pin down in this paper which other interneurons are responsible for that coordination between modules. What are the implications here for grid cell computation? The incomplete structured local connectivity is consistent with continuous attractor dynamics, where you have excitatory-inhibitory interactions between the stellate cells and PV plus interneurons that could generate the activity bumps underlying grid cell firing, though we'd have to look at a different paper to get a nice layout of that. The authors here note that this local circuitry is not well explained by grid cell modules that don't incorporate synaptic interactions, and argue that those models also fail to account for experimental evidence of attractor dynamics. Now, there are some limitations the authors already pointed out. Focal single photon optogenetics can't precisely identify which individual presynaptic neurons are activated due to axon dendrite activation. And throughput still remains a bottleneck for the procedures that they demonstrated here. And for that, the authors are calling for soma-targeted opsins and all optical approaches, which they think could improve circuit mapping resolution. Now, that's the end of the presentation of the paper itself. Of course, I was still feeling a little bit unsatisfied here because I didn't really get to see a computational model. But one of the things that jumped out at me here is that I think grid cells are still a very difficult candidate for a memory decoding challenge, to use for a memory decoding challenge. And there are multiple reasons for that. One of them is that it's still not clear how we would denote learned non-trivial memory in this structure. But another, as you can see from this paper and from the many open questions and the fact that this doesn't directly connect to a model, is that there's still many points of debate and unknowns involving the circuit models of input that's driving the transition of grid cell attractors during motion. There are multiple competing models. So yeah, that's my, I guess, initial verdict about grid cells and memory decoding. Now I'd just like to open this up to questions and discussion. I am done. Yes, go ahead, Ken.
That was great. I found this paper very, very interesting because it actually lays out what would be needed for a connectomic study. I just wanted to, this is more of a statement. It's like they did a lot of electrophysiology here purely to get connectivity. So this could have been a connectomic study. Maybe it would have been much more difficult, but like a connectomic study could have answered all of these way better, in my humble opinion.
Yeah, I suspect it could. That my observation, just from having been in labs like that, like this lab is closely related they work a lot with the Lisa Giacomo lab, and the Lisa Giacomo lab in turn works a lot with the Michael Hasselmo lab. And one of the things that has gotten really clear from being exposed to that is that there's a sort of a subset, a group of labs that they bet on functional recording, on electrophysiology wholesale. And then there's another set of labs completely separated from those that bet on connectomics. For some reason, I think there are some good overlap labs, but you do see this segregation, basically. I don't know if you've noticed that yourself.
And just and just one thing. I'm sorry. The other thing is like all of these things that you all of these studies that you covered in this paper, they were all on slice, like 400 micron slices or 300 micron slices, and that means that they didn't have recordings of what those cells were doing in terms of grid behavior. So it's like when we imagine a connectomic study, we imagine a study where you would have calcium imaging of the entire grid cell population that is then correlated with with connectomics of that same population. And this couldn't even answer if two of the stellate cells were in the same module or not. Right. So I agree. There's a complexity here, but this type of a study is just scratching the surface of kind of is the rough statistics of connectivity kind of in the ballpark of what these models would suggest.
I think you're absolutely right. And this separation in methods or preferred methods, I think in part comes from the fact that these labs have put a huge emphasis on very precise timing, on comparing phases of activity and models where you have nested oscillations and all of that stuff, which pops out best with electrophysiology. Calcium imaging is not really fast enough to capture very precise differences. And it's that legacy, I think, that then carries over to their attempt to do what really should be connectomic studies. Yeah, I agree with you on that. I'm seeing a couple more hands up, but at the same time, I am not seeing any motion. So am I online still?
Yeah, you're still there. I think Zach was first and then me.
All right, Zachary, go.
Okay, so I came a little late to the party, and I was doing a little catch up on the paper, and some of this was also over my head. But I did want to ask. So it says on that second bullet there that the local circuitry is not explained by grid cell models that don't incorporate synaptic interactions. Can you talk a little? What does that mean specifically? Like synaptic interactions? Do they mean like shunting inhibition type interactions, or?
No, I think they're just talking about the interactions between local versus non-local synapses, the interaction, being able to observe synchronicity versus not synchronicity. So that's what I think, but I don't remember exactly from the paper, so I'm not 100% sure. Maybe someone else here picked up on that. Someone want to jump in on this? No.
Okay. Yeah, I was just wondering if they meant specific other than like connectivity. But okay, all good. Thank you.
Ariel.
Yeah, I was thinking while we were going through this paper about the different spatial scales you can have grid cells at. Like you have ones that are more tightly packed and ones that are have broader spacing when an animal's moving through its environment. And thinking about what they were showing here with regards to the different changes along the like dorsoventral axis, because you mentioned up the front that you tend to see the bigger versus smaller ones at different positions along the dorsoventral axis. And it wasn't obvious to me that there's a clear like one-to-one sort of explanation of, well, you see these changes in physiological properties going from dorsal to ventral, and that's probably going to correspond to bigger or smaller grid cells. But I'm just wondering if I might have missed it, and you think there's a cleaner explanation here, or there's still more work that needs to be done.
Maybe I'm not really understanding what you're saying, because what they're saying here is that if you track the dorsoventral axis and you have cells that are at a close distance, that they're going to be more synchronized. They may belong to the same module or not, or even the same cluster, and there can be clear boundaries between them. And so as you're going through the dorsoventral structure, if you're further apart, then it's likely that you belong to a different submodule, which makes sense if one set of grid cells is giving you small grids and the other is giving you large grids. But maybe I'm totally misunderstanding what you're trying to say here.
Yeah, no, I'm trying to work out myself. So let's say you're going into an area that has larger, like a grid cell, where it's corresponding to a larger spatial area.
Yeah.
Then you would expect. I'm just trying to work through what you would expect in terms of like the physiology to be different. So an animal, that means like as the animal's moving around, that cell should, maybe I'm confused about what it means to have grid cells with larger phases and larger spacing. Does that mean that the animal, when it's moving through its environment, that particular grid cell is firing for longer, but then there's also longer gaps between?
Yeah, there's longer gaps as well. You have a greater spatial sort of grid, meaning that you'll have firing at larger distances between the points where the grid is most active.
Yeah, okay, so I do have that. Yeah, I'm just still confused as to whether basically this paper says anything about why that might become the case as you move dorsal to ventral, or whether it's orthogonal to those considerations. Maybe Ken.
I guess Ken has the answer.
Well, I probably don't have the answer, but this dovetails into the question that I was going to ask. So if that's okay, I mean, all of this comes down to, from the model point of view, all this comes down to the cells that move the bump, right? And so you can have exactly the same bump attractor with exactly the same cells that move that bump around. But if those cells are driven by the little foot movements in a different way, then you'll get a different spatial scale, right?
Yeah, that makes sense.
And so my question, I was hoping to get Randal or anybody else to speculate a little bit more on. So I had not realized that there was that the stellate cells and the pyramidal cells have different interneuron populations that they're getting their stuff from. Do you feel comfortable with laying out some speculation on how bump attractor is being moved around, or how this actually maps onto circuits or anything like that?
If you're asking me directly, no. I briefly browsed through the Hasselmuth paper that had an actual nice model of generating attractors, generating from activity at different phases and all that with the different cells and how they're feeding information to one another. But I didn't dive in deep enough to be able to remember how that works. So, I don't really want to do a third paper on grid cells since we've already covered so much in grid cells. But I kind of wish I had picked that paper to give a model instead of giving this anatomical, say, function to this is funny because this is function to structure rather than structure to function, what we usually talk about. That's what this paper is trying to do. But I kind of wish I had. So the more I was digging into this paper, the more I thought, I wish I could present the Hasselmuth paper instead. But maybe someone else here has the grid cell model in mind, maybe recently done or so.
There's a paper that I'm reading that came out of Genelia recently, a bioarchive paper on the head direction cells in fruit fly, and they are essentially trying to, because they got the full connectivity, right? And if I'm understanding it correctly, the reason why I'm bringing this up is because I feel like the grid cells are not overly complex, but they're more complex. There's less known about them. They don't have the full connectivity. And so we could look at a model where we do have the full connectivity and we do have calcium imaging, and they still don't understand what the hell is going on. And basically what I was gleaning from this, which is a theoretical paper again on bioRxiv, is they were like, look, all of these models suggest that the connectivity for a bump attractor would have to be so precise in order to not have it like lock on to act as a continuous bump attractor, right? And they were trying to address that problem, comparing it with the connectivity that they get in the connectome, which looks to not be anywhere close to precise enough to give a continuous attractor. And so it seems to me like even in the simpler systems, when they have the connectome, they're still not able to connect the dots, and they're trying some very novel things to try to get to reconcile those.
It does sound to me like that would be a more interesting system to dive into at the moment, just because it is a little bit simpler and you have more data.
And it has the
yeah. Okay. So we can get back to attractors again, but this time with a simpler system. Yeah, I think that might be a good idea. Any other questions, thoughts, observations before we call it a day for the session? All right, if not, then thank you everybody who joined us. As I just want to remind everyone that we have these memory decoding journal club sessions every 2 weeks. And we've been spending a lot of time going through papers that we are selecting as maybe examples of an animal model that would be a useful animal model in which to describe a proposal of how a good memory decoding experiment could be carried out and published. We haven't always done that in the past. We've focused mostly on presenting new papers that are candidates for winning a memory decoding award or potentially even the prize. And I think as the year goes on, we're going to refocus to presenting those types of paper. Although perhaps, I don't know, maybe in the next couple of weeks, we'll still be going through a few animal models potentially. It's just so that you all have a bit of a, you can look ahead and expect some expectation for what we might do here. But it's every 2 weeks, and I'm very grateful for everyone who showed up. Thank you.
Thanks, Randal.
All right. Bye-bye, everyone.
Thanks.